Thank you. It's really a pleasure to be here and give you my perspective after a quarter of a century with this disease now. Starting next year, it'll be 25 years that I've been involved in this. And while I sense a lot of angst and frustration amongst the patients, I can tell you there's some forward progress being made, because when I first was here in 1988 in Oxford, they had 82 presentations by psychiatrists and 2 by other physicians, and that's a true historical fact. So there has been some progress since 1988, even in this country. I was asked to talk about my perspective of past, present, and future in 15 minutes, and that's really quite a task. But I think I've summarized it, and hopefully it'll be of some benefit to you, particularly perhaps in the direction that we may be going in this disease process. Well, of course, you know, it's an illness of many, many names, and actually, historically, the disease process probably first got, quotes, named and described here as ME. In the U.S., we've had a big problem with that because we sit and technologically argue that there's been no demonstrated itis, so we can't have something that has an itis in it. So that's really what's happened with the holdup there. Of course, as you probably know, at the latest meetings in the U.S., there was a ad hoc committee that decided perhaps we should merge these together and start referring to it in the literature as CFS/ME and try to bridge these gaps in terminology, which really I think has caused tremendous problems over the years. My associate Paul Cheney and I are responsible for yuppie flu, and I apologize for that. And how it happened was in 1984, when we had people who got very, very sick from which they didn't recover. They were labeled yuppies because we practiced in a very affluent area of mostly white people, and they had the flu that didn't go away. So there came that terrible name. It was coined actually by Newsweek magazine. It was nothing scientific about it, and they did a cover on it, and it stuck for many, many years, and I think caused a lot of grief and problems. I was then part of the group that was formed by the CDC to perhaps bring some clarity, at least of definition, to this. It was an unfortunate naming experience because we ended up talking about symptoms. The meeting dragged on, and finally, at the end of 2 days, everybody was tired of it. We said the only thing we could agree upon is chronic fatigue syndrome. And that, unfortunately, is the nomenclature that's been used in the United States for a long, long time. You know, as a result of that, the 1988 Holmes criteria were developed, which were basically research criteria which actually looked preferentially at people who had post-infectious fatigue states. And so that definition is somewhat biased. It was revised in 1994, called the Fukuda criteria, which have a lot of problems as well, and I won't get into that, but I will say that most practicing American physicians in this disease now have adopted the Canadian consensus definition that I think is a very useful document, particularly for practicing physicians, and I would encourage all of you patients and support groups, et cetera, to try to get similar guidelines adopted here. And I know Byron has worked on it some, and there's a lot of work out there that should be paid attention to so that we can worldwide use at least the same definition for what we're talking about. I'll take an aside to say that because of the political pressures and problems we have with this disease, in forming the Institute, we chose to not use any nomenclature except the more generic neuroimmune disease because it avoids that politicizing that immediately happens when you use ME or CFS. There were attempts, of course, to be more specific. In Japan, for a long time, this was called low natural killer cell function because all their patients had low natural killer cell activity and numbers. And that is still a very useful marker, but it's not very useful for a name of a disease, of course. And then chronic fatigue immune dysfunction syndrome, which we had this ongoing debate in the U.S. for years, especially at the governmental level, in that it was a feeling that not all the patients had immune dysfunction, and sometimes immune dysfunction may have not been the major feature, and it still trivializes it with chronic fatigue. So as far as the name goes, I don't think we're going to make much forward progress until we know more about the pathogenesis and the subsetting, et cetera. So we're sort of stuck with what we've got, along with all the political problems that come to bear with that, unfortunately. So I want to talk about a couple of myths that still exist here in the United States. One is that it's rare and who cares about it. Well, it's not particularly rare, as you've heard from every single speaker here. By conservative estimates, we have 500,000 people in the United States, and that's very, very conservative estimates from physician practices. It doesn't address the tip of the iceberg. It doesn't address the people who are self-diagnosed or not diagnosed. And so locally, it gives me a population of about 6,000 to 12,000 people, which is really a pretty significant disease. As you've heard before, the non-medical burden in the United States is $9 billion a year. They say if you add the medical burden to that, it's probably about $20 billion. So that does get attention of politicians. They do look at numbers like this, and they look at market potential, et cetera. The estimated market potential of a diagnostic market in the United States is felt to be about $100 million a year if everybody who complained of fatigue just had one test. So there should be interest in terms of the biotech industry and that sort of figure. And if there were a therapeutic intervention, the estimated return for a drug company would be supposedly about $3 billion a year if there were an effective therapy that took 3 months to administer, just on an average. The second myth is that it's prevalent amongst young, affluent, white professionals who have nothing better to do but complain and visit their doctors. And in fact, women, men, and children are all affected by chronic fatigue syndrome, as you all know. It's often misrepresented as only a woman's disease, and I feel kind of bad for the men who are clearly diagnosed because it's really even a more orphan disease for them, and I think the suffering is sometimes greater. And the statistics, this was one of our earlier studies that really showed not quite as disproportionate amount as other centers have reported. This was a surprise to me until it was duplicated in California when Lenny Jason first came out with this and found double the prevalence in Latinos. And I think we simply weren't looking at originally in the original studies. These are strange populations. You have to get into the inner cities to really find it. The same with the Afro-American. It's always questioned in our federal grants why we don't address American Indians. And they're very hard to find. And they're serviced through a special division of the health services, and nobody within that system is very interested in this. And so we show the incidence as zero, but it's probably not true. It probably simply hasn't been looked at addressing your concerns earlier. I think the most overlooked population is the kids. And as an internist, I don't see people under 13 myself, but I think it's widely undiagnosed, and I am very concerned about this problem because it's a really hard one to address. Very commonly in the United States, they're labeled school phobia and put in the system of behavior modification rather than diagnosis. And I think the shame of that on top of everything else is that it has such an impact on their educational performance in their life. It's bad enough to suffer this disease when you're 40 or 50, but during the time when you're missing school, missing social activities, et cetera. Here's a picture of one of my patients who was placed against his will in a psychiatric hospital for 2 years and received electroshock therapy, amongst other things, prior to my seeing him. So that's another problem that I think has to be addressed, maybe as a separate issue. I know David Bell has very strong feelings about that in terms of how that should be handled. The third myth is that CFS is a form of depression. And this is a historical argument that went on for many, many years in the United States. And so, as my style would have it, I took this on with a federally funded NIH R01 grant to either prove or disprove this theory that chronic fatigue was a form of depression. And in fact, most practicing physicians know the two entities are very, very different in terms of onset, incidence, symptoms, attributions, lab tests, et cetera. And even now we know in terms of gene expression. But I wanted to demonstrate it simply laboratorily if I could. And in fact, if I had the time, I'd show all the slides that you can differentiate on SF36s and all kinds of other functional studies. And clearly, depressed patients turn out differently. But I just will show this one summary slide in which we put 5 immune variables, and we used RNA cell, NK cell function, and I think the cytokines. And In any case, and this was a 66 control, 66 depressed, and 66 chronic fatigue patients. And you can see when we did multivariate analysis, it was very, very easy to differentiate the chronic fatigue patients from both of those control groups. And actually, we could use immunological markers to separate normal controls from depressed controls, which is an interesting observation as well. But there was very little overlap, and I think this shows that there are biological markers that at least can very easily differentiate chronic fatigue from healthy controls and from depressed controls. That isn't terribly costly and is pretty well documented. Now, part of this confusion comes from the symptom and defining a disease by symptoms. And if you look at this whole array and the frequency and everything, it's why primary care docs hit themselves in the head with this. Like, this person is complaining of 40 things, and I don't understand it, it doesn't fit a pattern, I don't like it, and it's just, don't bring me your laundry list kind of thing. And I've been faced by that same kind of problem. You look at it, it's absolutely stunning. And once we got to thinking about this, you thought, well, that has to be a reason for that. And I think what the world has come to view, the people who are in this field, is that every system, it's a total body disease process for whatever reason, whatever's driving it. And what we've come to realize is that depending on which part of the elephant you were looking at or feeling, you had a different bias and a different perspective on this. We certainly know the rheumatologists have their fibromyalgia and their very strict definition of fibromyalgia and what determines a fibromyalgic patient and what doesn't. We know the gastroenterologist now will diagnose leaky gut syndrome, but only within very careful parameters and not looking at the whole person. The autonomic abnormalities with orthostatic intolerance and resting tachycardia, et cetera, has drawn the attention of a number of centers. And they believe that that's the crux of the disease process. And if you only fix that fix the low blood volume or whatever the defect was, you'd make these people well, although that hasn't turned out to be the case. The HPA axis has gotten a lot of attention in the United States, and those abnormalities have been well documented. Nobody disputes it. Even the gene array studies now document abnormalities of the HPA axis. So I think we know that those abnormalities exist. The question is what to do about them and where they came from and how to put this all together in a piece. I've been particularly interested in the immune derangements because that's where I started, with evidence of viral reactivation with the herpes viruses, and many of you know the whole story of HHV6 and how it was originally isolated from patients in the Lake Tahoe epidemic. So clearly, and this is where we came up with the idea of the Institute, because how could one physician, particularly a GP kind of physician, which in the United States now the average visit to your GP is 3 minutes in duration. So how are you going to possibly address a disease like this in 3 minutes? Well, the answer is it's impossible. So we looked for a different model, and I said, how can I possibly ever deal with this? And that's where I came up with the concept of an institute where we will bring in these specialists to see you. So if you have a problem with orthostatic intolerance, you may see that cardiologist who deals in that and is coming in to visit the clinic once a month and knows about it and knows about ME/chronic fatigue syndrome, so we're not reinventing the wheel, you're not in an adversarial situation, et cetera. And I might make the side that the worst possible place for a chronic fatigue patient to show up is in the emergency room, because they get totally bad care, totally misunderstood, and totally put down. So it's a very negative experience for most patients to end up there. So there's a proposed model that there's some disagreement on this, and Kenny may redraw my lines in different ways, and other people in the audience may as well, and they may put double lines some places and X's other places, but basically I think we know that there's immune dysregulation, and as a result probably of genetic predisposition, environmental factors including stress, chemical exposures, and infections play a role there. If they're not causative, they're a consequence. I am convinced of that. Whether it be bacterial, viral, fungal, or parasitic, you get this immune system breakdown with multiple chemical sensitivities, chronic fatigue syndrome, multiple allergies, et cetera. And there's a lot of support for this that's in the literature now, is peer-reviewed, and it has been published. So the allegation that there's no good science out there is simply not true any longer. I agree that originally it was very anecdotal. Well, a lot of the studies were not controlled, but that has largely been corrected. So past research that I've been involved in has been looking at the the immune system, particularly the T cell function, the decreased function of the natural killer cells, the abnormal RNA cell function, abnormal 2,5-A binding, and the dysregulated pro-inflammatory cytokines, because I think that represents a very definable, identifiable subset. Now, I also often get asked what percentage of the total CFS population has these abnormalities, and it's about 70% in my practice. So there's still a group of people that don't have this exact model, don't have these exact abnormalities, or don't have the complete panel. And this was based on the bias that infectious agents may trigger and perpetuate chronic fatigue syndrome. We certainly have around the world epidemics of chronic fatigue syndrome directly related to infectious agents. The Australians have been particularly good about demonstrating this and have even demonstrated proactively. They've gone to where there was an acute outbreak of an infection like Ross River virus and followed it forward to see how many people develop chronic fatigue syndrome. So certainly that's a subset. And we know that the herpes viruses can be involved. A number of studies have shown reactivated herpes viruses, particularly 6A, which is particularly neurotrophic. There's other very good studies showing evidence that the disease process can follow parvovirus infection, enterovirus infections, et cetera, and mycoplasma. I couldn't agree with Kenny more with respect to that particular finding. And our current research that's going on at the Institute right now is basically assembling and organizing our repository and database. We're doing gene expression profiling, very similar to the previous speaker, virus expression profiling as well, and proteomic profiles. And I'll just tell you a bit about our repository because I think it's very important, and it's something that could be done here fairly cheaply and fairly easily and will contribute a great deal to science. Currently, we have 5,000 samples that are fully characterized with a searchable database. If you want a 40-year-old woman who has low NK cell function, who's been sick for 10 years, we can search for that. Our particular freezer that was provided by a local support group, can store up to 25,000 samples at -80. And I was lucky to start banking specimens in 1984, and many of them were aliquoted back then just because I didn't know what I was doing. So most of them have never been unfrozen. And they've been at -80. I was just waiting for the test to come along. And what we have is over 3,500 serums, 130 spinal fluid specimens, over 500 PBMCs, et cetera, et cetera. I often get asked the question about donation of bodies. And we don't have a mechanism to accommodate that request right now, but it's come from around the world, and I think it's something that the support groups should and are capable of taking on and address this issue, because that is a very valuable resource. It's a hard fight now because the coroners don't want to do it. It costs money, and they'll say, let's say it's a suicide, they say cause of death is gunshot wound. So we know nothing about the underlying pathology, unfortunately. I think that's something that internationally we could take on and really accomplish. Here's the protocol if someone wants to donate to science, and here's how it happens, and here's where the specimens are going to be handled and stored, et cetera. So we've started collaboration also internationally for some of these studies because we recognize that we've been guilty of this looking at one thing at one site rather than collaborating, particularly worldwide, and taking advantage of people who have a great deal of interest and expertise in this disease process. And you've already heard about how gene and virus expression and profiling is done, but patients are always sort of fearful about this because they think there's some voodoo science here, that they're looking at their DNA and RNA and things like that. And so I often explain to patients that expression, I think, can be really useful. Its resting state means something. Its stimulated state may mean more after an infection. We're doing an interesting small pilot study right now about what happens to gene expression after sequential treadmills in chronic fatigue patients. So we're exercising them 2 days in a row, and the second day about kills them puts them in bed for weeks and weeks and weeks, but we want to see if we elicit a different gene response after that, because there's some preliminary evidence that you do. Because one of the, of course, critical features of chronic fatigue is that post-exertional malaise, whether it's mentally induced or physically induced, and exactly what is causing that. We hope that these gene array studies may be able to answer that at least, and there's some evidence that it does, that we can point to the mitochondrial defect or whatever the defect may be. This is a CDC slide that I just showed to show the utility of the genetic studies. And this, I think they took 39 genes here. This is Susan Vernon's work, and looked at them and said, could we identify the non-fatigued patients from the chronic fatigue patients? And indeed, their conclusion was that can fairly safely be done. There is this group in the middle that is also different, but doesn't meet all the criteria for chronic fatigue syndrome. That's what's labeled ISF. And then we started virus microarray results, as you heard a previous speaker, and we sort of ended up doing this somewhat simultaneously here. Part of that was because the technology is now available. And we found some similarities between the UK patients and the US and some differences, which is very, very interesting, very predictable, because it may well be that this is regional in terms of what virus is activated or is causing a disease process. And interestingly, the patients had a very high viral load versus the controls. And what we're in the process of doing now is repeating it a second time so that we can wash out the acute influenzas and rhinoviruses and things like that we found as background noise. But very often a chronic fatigue patient had evidence of 40 or 50 different viruses being active versus the controls that were about 2 to 3. So one of the things we're involved, one of our primary projects here is running the PCRs and things to confirm. What we're doing is we started with the top 50 hits out of those 1,600 and to see if there's any real commonality that comes through on this particular patients. And I will be speaking some more about that tomorrow. We're looking at the cytokine and proteomic profiles by another array. This generates a tremendous amount of data, and it really requires a sophisticated analysis in order to make heads or tails out of these kind of profiles. The chromosomal integration of HHV6 is a project we're doing in collaboration with Lucinda Bateman to see if perhaps some of the patients have chromosomally integrated HHV6 as part of their disease process, and it seems to be that a subset does have that problem. And then we're looking at the mechanism of the NK cell dysfunction. As many of you are aware now, we can subset NK cells into the bright and dim NK cells and try to figure out exactly what their role is and why their function is so low. The interesting thing about NK cell function is that if you treat a patient and they improve, their NK cell function improves. So it's a fairly good marker of their status. Another project we have is this issue of HHV6 latency, because most herpes viruses should go into latency and should only be reactivated at certain times. So why they're reactivated in chronic fatigue syndrome. And then our last project that we have on the books at the present time is our studies on epigenetic dysregulation. So thank you very much.